This is chapter 3, the cell, and cells are considered the smallest living units of life. And we will start off talking a little bit about cell theory. And cell theory, like we said, the cell is the structural and functional unit of life. All organismal functions depend on individual and collective cell functions. Biochemical activities of cells dictated by their shapes or forms and specific subcellular structures, which we usually refer to as their organelles. And in order for life to continue, cells have to perform their roles and divide and carry on life as it exists. Now, this is in this hierarchy that we mentioned in chapter 1. The last chapter, chapter 2, we started with the beginning of the hierarchy, speaking to atoms, and atoms being put together to form molecules. Now we're looking at the next step in this, looking at organelles and cells. So we're building a bigger picture from the smaller building blocks. This is a picture that's going to show you different types of cells. There's a lot of diversity in what cells look like, and a lot of it depends on what their operation and what their role is in the body. If they are transporting things and they have to go through small passageways, round is a good shape to be able to not get hung up in the blood vessels. So you see that the erythrocytes, red blood cells, are round. Epithelial cells are packed pretty tightly together. Epithelial tissue is found in the skin, and so they have to be tightly bound together to make a good boundary against substances from the outside getting into the body. These are fibroblasts that lay down collagen fibers that make up connective tissue. Here's your muscle tissue cells. They're long. They have to contract and shorten. Fat cells store a lot of fat, so they have a little vesicle called a fat globule that stores the fat there. Macrophages are cells that fight disease, and they operate and move by amoeboid motion, so they have to make extensions to be able to move around from place to place. Nerve cells kind of look like they have a long wire that's separated off from the nucleus part of the cell, and they carry information from place to place. So that's the processes that you see that transports electrical impulses from one part of the body to the other. And then some cells have a tail so they can be mobile, and in the body there's only one, and that's the sperm cell that has a very long tail called a flagellus. So these are just some examples, but there are over 200 different types of cells found in the human body. This is a generic cell, or a generalized cell they call it here, showing you all of the major features. So as you go through studying the cell and preparing for lab quizzes, you want to get familiar with all the various organelles and what they look like on this picture. And as we go through, we will talk about these different organelles and what their functions are. So the cell has three major parts. Many parts within this three-part breakdown, but these are the three major parts. The boundary around the cell is called the plasma membrane. And then within the plasma membrane, there is a liquid-based medium that's called the cytoplasm. And if it's something inside the cell, it's called intracellular. So the cytoplasm represents the intracellular fluid. The organelles are the little miniature organs that are suspended in this fluid called the cytoplasm. So the plasma membrane is the outer boundary. It separates the intracellular fluid, the fluid that's inside the cell, from the extracellular fluid, the fluid that is outside the cell. Now, membrane lipids are mostly phospholipids, and the membrane is considered selectively permeable, meaning that this boundary has the ability to control what gets in and out of the cell. Now, besides the phospholipids, which make up 75 to 80% of the cell membrane lipid component, you also have some cholesterol, about 15 to 20%, and that's a stabilizing feature in the membrane. There are membrane proteins embedded in this phospholipid bilayer. And the integral proteins are proteins that go all the way through the membrane. And these function as transport proteins, channels or carriers, enzymes or receptors, and we'll talk a little bit about these in a minute. And then you have these proteins that are either located just specifically on the outer face of this plasma membrane or on the inner surface. And these function as enzymes, motor proteins, or they make cellular connections. So they can make cells snap together, or the ones that are on the inside, they can actually anchor fibers that make up the cytoskeleton. Yes, the cell has its own little skeleton, and we'll talk about that a little bit later on too. Here's a picture of the plasma membrane. There's a little small view of that cell we looked at earlier. And this is this outside boundary. And you can see, if you remember back in chapter 2, we showed a picture of the phospholipid. Now the phospholipid has a phosphate group. These little round circles here in lighter blue are the phosphate heads that has a charge. The lipid part are the tails that protrude into the middle, and these are fats, and they're nonpolar. They don't have a charge. So part of this molecule is a part that's polar with a charge, and the other part is nonpolar with no charge. Now the part that is charged is called hydrophilic, meaning it loves water and it can merge with this water boundary, this water-based fluid on the outside of the cell. And these hydrophobic tails, nonpolar tails that are lipids, hydrophobic means water-fearing. So they kind of shy away from water. And just like if you have oil and vinegar, the oil is hydrophobic, it's water-fearing, and it doesn't dissolve in water. And you can shake up oil and vinegar, but it will still separate out because the fat and the water-based solutions don't mix very well. So the lipids will dissolve in the lipids. So if you have another layer here, see how it's opposite the arrangement of this one? Here now your phosphate polar heads face the inside, and the inside is a water-based environment too. This is the intracellular fluid. And your tails, which are hydrophobic or water-fearing, they dissolve in each other. So if you look at this, you have these oil, fat tails, okay? And they resist the water, and they make a barrier in the middle. And then your polar heads, outside and inside, they dissolve in the water-based environment. So you've got this perfect little lipid bubble that keeps anything that's water-soluble out and anything that's water-soluble in. So the only way that things can get through the membrane is through these proteins. And these proteins represent channels or carriers that will allow things that are not lipid-soluble, and most things are not lipid-soluble, to get into the membrane. So the cell can control them by controlling what comes through these different proteins. Now you can see there are these little, look like trees on the outside. These could be identification markers. They could be markers that will be attachment points for their cells. Here's one of these peripheral proteins we were talking about earlier. They're on the outside or on the inside. This could actually connect a cell to another cell. This one here can anchor parts of the cytoskeleton, and the ones that go all the way through are called integral proteins, okay? So that's just an idea of how the cell membrane is designed. It's a biphospholipid layer, okay? And these phospholipids are both hydrophilic, with the polar phosphate head with a charge, and partially hydrophobic, which is nonpolar, and this is the lipid tails. Now, we said that these proteins can work as transport. So I'm going to give you a couple of examples. There's many different ways that these proteins that go through the cell membrane can work. But here's an example of where a protein that spans the membrane may provide a hydrophilic channel. Because remember, the membrane's hydrophobic, okay? You can't get through here. But this provides a channel where molecules that are hydrophilic, water-loving, they can go through that particular channel. Now, some transport proteins that use ATP as an energy source, ATP, remember, is like the gasoline for the cells, and they can actively pump the substances across the membrane. So sometimes it's just a passive diffusion, and sometimes it has to requires a little bit of energy. And here's an example of looking at intercellular joining. Some of these proteins actually can link cells together like Velcro. And CAMs are cell adhesion molecules. So membrane proteins of adjacent cells may be hooked together in various kinds of intercellular junctions. Some membrane proteins, cell adhesion molecules, or CAMs, this group provide temporary binding sites that guide cells migration and other cell-to-cell interactions. So they're used to hook cells together. Now, cell junctions. So we just talked about cells being hooked together, but some cells are free. Blood cells are free, sperm cells are free. They can travel from place to place. But some are bound into communities, and the way that they're bound together are 3 different ways. And the 3 different types, they're called tight junctions, desmosomes, and gap junctions. So let's look at these. If you have proteins that very tightly bind the cells together, this is the plasma membrane, and holds them together like Velcro, snaps very tightly together. This is a tight junction. It's an impermeable membrane junction, and it prevents molecules from passing through the intercellular space. So you can see them here. If you have little fibers, protein fibers, that allow a little give, they can slide past each other, this is a desmosome. And these anchors, anchoring junctions bind adjacent cells together. This says Velcro, but I don't think this was a Velcro like the first one. I think this is a poor word to use here because there's a little give here. These can actually collapse on each other and they can expand. So you have a little bit more room for movement between the cells. And where you have a lot of elasticity in tissues, you'll see a lot more desmosomes. A gap junction is like a channel. And see them here? So you have proteins that actually go all through both membranes so you can exchange information between the cells. And where you have cells working in a group, like in the heart, the atrial chambers, those cells all contract together at the same time. The ventricles, those cells all contract together at the same time. They're all connected by gap junctions. And this way they all beat together as a unit. Okay, so now we're going to look at types of membrane transport. Some of the membrane transport, like we mentioned on a previous slide, going through these channels doesn't require any energy, so that's passive. If it requires ATP, then that means that it's active. So again, passive processes don't require any ATP. And this substance moves down its concentration gradient, meaning that it moves from an area of high concentration to a place where it has a lower concentration. Active transport processes, ATP is required, and in order to have ATP, you have to have living cells because living cells make ATP, and many times it's going against the gradient to push substances where it is already high. So types of passive processes. You have diffusion. And diffusion is defined as the movement of substances, usually solute, from a region of high concentration to a region of low concentration down a concentration gradient. So simple diffusion is just that. The solute moves through the membrane, is small enough to get through. If it's too big and it's not lipid soluble, then you need a carrier. And we saw a picture of that earlier, a channel that's hydrophilic that allows the substance through. Or you can have a carrier. And a carrier is something that opens and closes in response to something binding to it. Could be a hormone, could be some kind of a cofactor that has to take place, co-reaction that takes place before the channel will open. So you either have channel or carrier mediated. These are proteins. And because the proteins act as a facilitator, using a channel or a carrier is called facilitated diffusion because you need something. You needed this protein to allow this substance to get in. The other type of simple diffusion is osmosis. And osmosis involves the movement of water. So we're going to talk about these a little bit more in more detail. But for now, you have diffusion, and filtration is driven by differences in hydrostatic pressure on two sides of a membrane, and we're going to cover this in A&P2. So just know that it is a passive process, but I'm not going to speak to it in any detail here. This is a look at carrier-mediated facilitated diffusion, where you have a specific protein carrier that will allow one chemical to get through, and there's actually binding sites that allow this substance to enter and exit the cell. Now this is looking at a channel where you have small lipid-insoluble solutes that are very tiny, and they can get through the channel, and you can see they're higher concentration here, lower here. So the definition of diffusion is the movement of solute. These are solute molecules that are moving from higher concentration to lower concentration, and this is facilitated diffusion because the facilitator is the protein channel. Now, osmosis is the movement of water. Now let's just back up here just a little bit because I want to review something. When we're saying solute, we're saying the substance is in lower quantity. Remember when we talked about what a solution is made up of? A solution has two parts, the solute and the solvent. When we're talking about diffusion and facilitated diffusion, we're talking about the movement of solute. Now we're talking about the movement of solvent. And solvent, the universal solvent, is water. So osmosis is unique from the other types of diffusion we just talked about because what's diffusing here is the water. So you want to make sure you keep that separate in your mind, okay? Diffusion, facilitated diffusion, those two things involve solute, salts, glucose, larger particles. This is specifically water when you're talking about osmosis. The definition of osmosis is the movement of water from a region of low concentration of solute, okay, to a high concentration of solute through a semi-permeable membrane. So you want to think about that. It's the movement of the water. The water is going to move to where the solute load is highest. So here's how osmosis works. And I have posted up some videos of, or one at least, on how osmosis works. So you can look at it, and there's many more on YouTube. But what happens here is if you have a U-tube, okay, and the water level is equal on both sides, and the water can move freely, but the solute cannot. Now the solute's the little red balls you see here, okay? Notice how there's more red balls on this side than there is on this side. So this is in the beginning, okay? This is how we start. So over time, what's going to happen is that you're going to have, oh, I'm sorry, take that back. There's equal distribution, they can get through freely permeable membrane, okay? Sorry about that. The membrane is freely permeable in this example. The next one is going to be the one where it's a little bit different. So what's going to happen here is that the balls can move and the water can move. So over time, what's going to happen is you get equal distribution. See how there's equal space in between the red balls and the water? And that's what you get when you have a freely permeable membrane, okay? So the end result is equal distribution, water levels the same, and the space between the red balls and the water is equal. All right, now let's go to the next example. In this example, you have the beginning condition, okay, where you have more red balls over here, fewer over here, but now you have a selectively permeable membrane. See, the holes are smaller here, so the red balls can't get through. So what happens is, if the red balls can't get through, then over time, the water moves through to separate the red balls out, so the water distribution to how many red balls you have is equal. So remember, the definition of osmosis is the movement of the water from a region of low concentration of solute to a higher concentration of solute, which you see that's what it's done here, through a semi or selectively permeable membrane that's permeable to water but not permeable to the solutes. So the word tonicity means the ability of a solution to alter a cell's water volume. So there's a couple of words that are associated with this term's tonicity. The term isotonic means that the solution with the same non-penetrating solute concentration as the cytosol, meaning that the solute load on both sides of the membrane is the same. Okay, that's isotonic. And if you were going to give somebody a solution intravenously that you want to be isotonic with their plasma, if you want to give them, say, a glucose or a dextrose solution, it has to be 5%. 5% is isotonic with what is in the plasma. So that's just one example. Now, if you have a solution with higher non-penetrating solute concentration than what's inside the cell, what's going to happen is that the water, okay, is going to leave the cell and go out into the area where the solute concentration is higher. So I'll show you a picture of that. If it's hypotonic and the solute load outside the cell is lower than what's outside the cell, hypo means the cell will blow because the water is going to come in and it's going to go to where the solute load is the highest. So hypo, the cell is going to blow up. And if it's hypertonic, if the solute load is too high, the cell is going to shrivel up. So here's some pictures of red blood cells in different environments. What you would like to have is you'd like to have your red blood cells in an isotonic solution where the solute load out here and in here is exactly the same. And see, the water can go in and out equally, and the cells retain their normal size and shape in isotonic solution because the solute load is equal. There's no unequal movement. Now, if you have too much solute out here, then what happens is, by osmosis, the water goes from a region of low concentration of solute to high concentration of solute, so the water is going to go out. Just like if you have a concentrated saline solution, the cells will shrivel up and they'll lose water. That's a hypertonic solution. In a hypotonic solution, what it means is that less solute out here, more in here. So osmosis means the movement of water from a region of low concentration of solute to high concentration of solute. So the water goes in and eventually the cells will burst. So the term for, I want you to know a couple of terms. In the clinical lab, if a cell loses water, it's called crenation. C-R-E-N-A-T-I-O-N, crenation. If a cell takes on water and it bursts, the cells explode, this is called hemolysis, H-E-M-O-L-Y-S-I-S. So those are two terms that I want you to know that are associated with hypertonic solutions versus hypotonic solutions. All right, now let's speak a little bit about active transport processes. There are two types of active transport processes. One is active transport itself, okay, using a active transport channel, and the other one is vesicular transport using a vesicle, okay. So one uses a channel or a carrier, and the other one uses a vesicle, which is a pinching in of the cell membrane to enclose a substance that's dissolved in the extracellular fluid, or it could be something that's too big to get through a channel or a carrier. Now, both of these processes require ATP to move solutes across a living plasma membrane because the solutes are too large for the channels. That's the case of vesicular transport. they're not lipid soluble, or the solute is not able to move down the concentration gradient. Earlier, I talked about active transport being involved with moving things against the gradient. So that's what we're talking about here. This is an example of a carrier that's called, it's a protein that's called the sodium-potassium ion pump. It requires ATP, and what you're doing is you're pushing 3 sodiums outside, and sodium is generally in much higher concentration outside the cell. So this is the extracellular fluid. So you're pumping sodium against the gradient, and you're bringing in 2 potassiums against the gradient because potassium is much higher inside the cell. This is the intracellular fluid. So pumping substances against the grain and using ATP to push sodium outside where it's already high, bringing potassium back in where it's already in. This mechanism here becomes very important in muscle contraction and passing along neurological impulses along nerve axons and conducting impulses there. For right now, I just want you to know that the sodium-potassium ion pump is an example of active transport using a carrier. Vesicular transport is using a vesicle. So like I said, if you have particles that are too big to get through a channel, you can pinch in the cell membrane, which is a biphospholipid membrane, and make a bubble, and this is endocytosis. And if there are things you want to get rid of from the cell, then that lipid bubble can fuse with the membrane and release the substances by exocytosis. So endo means in, so you're bringing substances into the cell. Exo means exit or out, and that's a way of exporting things out of the cell, and that's by using a vesicle. And it requires active transport. It requires ATP. All right, so now we have to talk a little bit about what's in the cytoplasm. We talked about the cell membrane being a biphospholipid layer, and now we have to talk about what's inside the cell. So the cytoplasm, I kind of call it the soup because it contains everything. So it's located between the plasma membrane, that boundary on the outside, and the nucleus, which is the center of the cell. It's composed of the water-based solution that everything floats in, okay? So it's basically water with solutes. So it could be protein, salts, sugars, and everything suspended in this water-based medium. The cytoplasm contains everything. It contains the organelles, the cytosol, and the inclusions. So I always call the cytoplasm the soup. The cytosol is the broth, because it's the water-based background that everything floats in. And the organelles are like all your noodles and your chicken and all your onions and carrots and all that stuff that you put in there, and it's a soup, okay? And cytoplasm is a bigger word, so you can always remember that cytoplasm is all-inclusive. Cytosol, smaller word, but cytosol is just a liquid water-based solution that everything else is suspended in. Now, your organelles represent the metabolic machinery of the cell, each with specialized function, and they're either membranous or they're non-membranous. And inclusions vary. Not all cells have inclusions. All cells have organelles, but not all cells have inclusions. So it could be like a lipid droplet that you find in a fat cell. You have macrophages, immune cells that contain vacuoles, things that they ingest, bacteria, things that they're trying to destroy. Some cells have crystals, glycogen granules, if they're metabolically active. And some cells have pigments, like skin cells have melanin that make pigment. Hair cells, skin cells. Hair cells have different colors, and so these are things that not every cell has, but only some have. Now, cytoplasmic organelles are divided into 2 major categories, whether they have a membrane or whether they don't have a membrane. And there's fewer that don't have membranes. And we're talking about the exact same membrane that you would find making up the barrier of the cell. So it's a biphospholipid layer. So the mitochondria, the peroxisomes, lysosomes, endoplasmic reticulum, Golgi apparatus, they all have these membranes. And then your cytoskeleton, centrioles, and ribosomes They don't have membranes, okay? And it allows for compartmentalization. And what this means is that when we talked about endo and exocytosis and we saw how things can move inside and outside the cell by these vesicles, these bubbles, well, things can move inside the cell too by bubbles pinching off and fusing onto different organelles and moving stuff around inside the cell. So that's really important. But we haven't talked about the organelles yet. Hopefully you had these in biology, and this will all come back. So let's go through the organelles quickly to finish up the chapter. So the mitochondrion we call the powerhouse because that's where ATP is made. And that's the really most important feature about the mitochondrion is that it makes ATP. So there's a lot of enzymes, there's a lot of inner membranes. When we get to chapter 25 or 26 down there at the end of AMP2, we're going to be talking about how that process of oxidizing glucose to make ATP occurs. So just remember it's the powerhouse, it's where ATP is produced. Ribosomes. Ribosomes are granules that contain protein and something called ribosomal RNA. This is where proteins are synthesized. Now, some ribosomes are free and they float around in the cytoplasm and they can synthesize soluble proteins that function inside that cytosol, that broth, or they can be used in other organelles. But then there are some ribosomes that are bound to the endoplasmic reticulum that synthesize proteins to be incorporated into membranes, lysosomes, or exported from the cell. So either they're free or they're bound to the endoplasmic reticulum, and that's what ER stands for there. So the endoplasmic reticulum is either rough, containing the ribosome, or it's smooth. Now, if it contains the ribosome, then it manufactures substances that have a protein component to them. Now, sometimes the endoplasmic reticulum that's rough can put together things like glycoproteins or proteoglycans or proteolipids, or you could have lipoproteins. So anything that has a protein structure with something else can be produced there. The smooth endoplasmic reticulum is a little bit different because it doesn't have the ribosomes. It's usually the site of lipid production. And it also has different functions in specific cells that are unique to those cells. In muscles, it's called the sarcoplasmic reticulum because it stores calcium. That's the function of the smooth endoplasmic reticulum. And you'll find that in things like, or cells like in the testes that make testosterone, there's a lot of smooth endoplasmic reticulum in interstitial cells because that particular lipid, testosterone, is made there. And in the liver has a lot of smooth endoplasmic reticulum that operates in detoxification processes. So you might see some different variations of smooth endoplasmic reticulum, but for now, I would just kind of abbreviate that and say they're important in the synthesis of lipids. And remember, with the rough endoplasmic reticulum, there's at least some protein component to whatever you're producing. This is the Golgi apparatus. Now, the Golgi apparatus is where things are packaged up. So your manufacturing plant is your endoplasmic reticulum. That's where things are made. And then through a vesicle, you can pinch off a vesicle and then fuse it over here onto the Golgi apparatus. And then through the Golgi, those substances can be packaged and shipped out. So, it's very much like a manufacturing plant with a shipping department. So here's your little vesicles that can be used to transport the substances from the Golgi after they're packaged to their final destination. So here's a look at the endoplasmic reticulum, showing you things working, vesicles being pinched off after the manufacturing's done, packaging, and you can see here showing you protein-containing vesicles pinched off the rough ER and migrate to fuse with the membrane of the Golgi apparatus. Proteins are modified within the Golgi compartments, and then they're packaged within different vesicle types depending on their ultimate destination. So they could be shipped out of the cell. They could be incorporated into the plasma membrane, like maybe some new membrane channel proteins or carriers embedded in there, something that the membrane needs. It could be also for lysosome production, and we haven't talked about lysosomes yet, but they contain digestive enzymes. They're made in the Golgi, so a lot of functions. So packaging plant is the Golgi apparatus. Now the next organelle is the peroxisome. And these are membranous sacs containing powerful oxidases and catalases. And what these do is these help to neutralize or detoxify substances that may otherwise damage or injure the cell. So catalysis means breaking down, being able to neutralize things that are would ordinarily be harmful to the cell, free radicals, highly reactive chemicals. And so these oxidases and catalases convert something that may be toxic into something that's harmless. So peroxisomes, detoxification of dangerous substances. Lysosomes. They look very similar to peroxisomes, but they're like the stomach of the cell because they contain digestive enzymes, and these are acid hydrolases. And because they're in the vesicles, they're enclosed, so they're in safe sites for intracellular digestion, so it wouldn't digest maybe things in the cell that you need and proteins that would be important for functioning of the cell. So you'd want them sequestered in a place where it wouldn't injure the cell. So it takes care of ingested bacteria, viruses, and toxins that are brought into the cell by vesicles. That's your vesicular transport. They can degrade non-functional organelles, break down and release glycogen. And also, if a cell is injured or if a tissue is non-useful, it can result in autolysis or apoptosis and cell suicide or programmed cell death when cells are no longer functioning. Now, the cytoskeleton is an elaborate series of rods throughout the cytosol. And they're proteins that link the rods to other structures. Remember, we talked about those peripheral proteins inside the cell membrane that can be used to anchor these cytoskeletal elements. But there's 3 types. And the smallest are the microfilaments. And an example of microfilaments would be actin and myosin. And these can be involved in changing the shape of the cells, especially in cells that move, or if you're going to make a vesicle and you're going to bring something into the cell through endocytosis. You have to create these little extensions of the cell to wrap around that substance you're bringing in and enclose it so you can create the vesicle. So those are microfilaments. Intermediate filaments help to suspend the nucleus in the center of the cell. So they radiate from the inner surface of the plasma membrane attached to anchoring proteins, peripheral proteins, and then they attach to the nucleus. And there are hundreds and thousands of these that extend from the cytoplasm and hold that very large nucleus in the center of the cell. Microtubules radiate across the cell from one side to the other, and they're involved with transport of organelles within the cell. They're organelles that have little motor proteins that allow them to propel themselves across these microtubules and move from one place to the other. So a very interesting network of fibers that are involved in making up this skeleton. Microfilaments, intermediate filaments, and microtubules. Now, centrosomes are an organelle that contains centrioles. Now, these are important in mitosis because the centrioles generate the microtubules and organize the mitotic spindle, and we'll talk about mitosis in lab. The centrioles, like I said, they contain these they're in pairs, these centrioles, and the centrioles can also form the basis of cilia, which are little hairs that extend from some cells, and flagella, which is the tail that you see in the sperm cells. There are cellular extensions. This is another thing that you see on some cells. Some cells have the short hairs, the cilia. Sperm cells, the only cell that has the tail. And then there's another type of extension that's called microvilli. Now, microvilli are finger-like extensions of cytoplasm that give the cell increased surface area. There's a picture of microvilli. And you can see having these extensions gives you a much greater surface area than if this thing was flat. Okay, so that's microvilli. Then we have the nucleus, and the nucleus is the control center of the cell. It's in the middle. It's surrounded by a double layer of phospholipids. So since it's a double layer of this bilayer, they call this the nuclear envelope. And it contains nuclei inside the nucleus. This is where ribosomes are synthesized. And the nucleus also contains the chromatin material that makes up the chromosomes. And that's where the DNA is that contains all of our genetic makeup. So this ends our discussion of Chapter 3.